Good afternoon. Welcome to the Tsinghua Week at Berkeley. This is an event sponsored by the Office of the Chancellor, organized and staffed by the Institute of East Asian Studies. My name is Wenxin Ye. I am Professor of History and Director of the Institute of East Asian Studies. It's my honor and pleasure this afternoon to serve as the timekeeper and facilitator of this opening ceremony. Our very first speaker is Chancellor Birgeneau, who will deliver his remarks now.
Good afternoon and welcome to Tsinghua Week at Berkeley. It's a pleasure to see so many people here today for this special occasion. I'd like to extend a warm welcome to the more than 130 faculty, administrators, staff, and students who have traveled from Tsinghua University in Beijing to participate in this week's events. Wan Ying. As many of you know, I had the honor to visit Tsinghua University in 2007, where I made the often quoted statement which also appeared on Central Chinese television, that Berkeley should be described as a people's university. Actually, in the United States, we use the phrase public university, but I think people's university is more appropriate, as we serve the public good for the people of California. In May 2008, Tsinghua University hosted us for Berkeley at Tsinghua, focusing on the theme of a partnership for excellence. Berkeley delegates participated in academic panels on topics ranging from history to international relations to nutritional science and toxicology. Our tennis teams also engaged in a friendship tennis match, while our men's octet entertained us all. This week, we are delighted to have the opportunity to act as a host to Tsinghua. I'm impressed with the many activities and events that are planned for Tsinghua Week at Berkeley. The richness of the program speaks to the extensive ties that have already been built by individual faculty members and campus academic units for exchange between our two universities. I'm impressed furthermore with the multidisciplinary nature of these exchanges that range from science, engineering and transportation, to architecture, public policy, psychology, philosophy, history, social welfare, and higher education. As you can see for yourself, the program over the course of the next 3 days ranges from research discussions between the Nobel laureates, a number of distinguished physicists, and one administrator from both campuses, to musical performances by undergraduates as they compare notes on their respective college lives. Panel topics for joint exploration range from urban environmental issues to technological and entrepreneurial innovation, from energy efficiency and green electronics to cultural psychology, from transportation safety and IT security to comparative political thinking in today's world. No fewer than 9 academic units have worked collaboratively to put together this impressive program, which will culminate in a closing panel that compares the systems of higher education in China and in the United States. As you are no doubt aware, a coordinated event like this is exceptional for a decentralized people's research university, which is far more accustomed to having each unit make its own international connections without necessarily notifying anyone else nearby. This week's program not only attests to the breadth and depth of Berkeley's overall strengths in multiple disciplines across the entire campus, it also demonstrates that we have the capacity to make internal connections within our own campus in the making of an international project that is of interest to faculty and students of multiple cohorts. In my 2005 address on Frontiers of Knowledge, Frontiers of Education, it's actually been reproduced in Chinese educational media, I spoke of Berkeley and its place on the Pacific, and of our looking from the Berkeley Hills towards the west, where the sun sets beyond the Golden Gate Bridge. Berkeley, in its founding vision in the 19th century, was to occupy the far edge of the western frontier. Berkeley, in the 21st century, is to occupy its place on the new frontiers of knowledge and education. It is to lead and guide the established world, creating knowledge about what we do not yet know. It is to build the kind of educational infrastructure that will enable a new generation of undergraduates to prepare themselves better for the life decisions that they are to make in an increasingly interdependent global order that characterizes the 21st century. The president of the University of Mexico once commented to me that in his view, the single most important skill for the 21st century that an undergraduate must learn in university is what he termed intercultural competence. It is only through an experience and an appreciation of other cultures that our students of today and citizens of tomorrow will be able to navigate through the globalizing world. Among Berkeley's hallmarks is an unmatched breadth and depth of academic programs, driven by a passion for inquiry and discovery, and marked by the integration and synergy of teaching and research. All of these characteristics will prepare us well, I believe, as we prepare our students for an internationalizing coming century. As we look beyond the Golden Gate Bridge that once framed the Western frontier, we see an emerging East Asia centered upon the momentous transformation of the Chinese world over the past 30 years. Berkeley's connection with China goes back to the 1980s when John Fryer, who sailed from Liverpool to China in the 1860s to advise the Chinese government on the learning of science and technology along with Christianity, left China for Berkeley so as to educate a new generation of Californians about China. Fryer was instrumental in founding today's Department of East Asian Language and Cultures, which is the oldest North American program in the teaching of East Asia. Fryer's personal collections laid the foundation of the C.V. Starr East Asian Library at Berkeley, which is the only standalone East Asian library building of significance outside of Asia. Under Fryer, the department grew to become home to many eminent scholars and students from all over the world, including China and Europe. Berkeley's contributions to China include notable Chinese alumni who became founding members of the Chinese Communist movement, the Chinese anarchist movement — we have a lot of anarchists at Berkeley — the Chinese cooperative movement, and the Chinese police force. In the 1950s and 60s, Berkeley was distinguished, on the other hand, by the presence of Professor Y.R. Chow, the leading Chinese linguist of the day, who previously had taught at Tsinghua University in Beijing in the 1920s and 30s. Berkeley has long held a position as a global university of consequence in East Asia. It has built strong ties with East Asian scholarly communities on a full range of subjects, from East Asian studies to earthquake and environmental studies to urban planning and architectural designs and much, much more. To extend our new frontier of intercultural competence and international knowledge, it is imperative that we strengthen the research infrastructure that connects the two sides of the Pacific. As a public university serving the public good, it is in addition imperative that we bequeath to the next generations enhanced educational opportunities that will prepare them better when facing life decisions in the globally interdependent new century. Tsinghua University, with its exceptional strengths in science and engineering and its new emphasis on English and Western studies is our natural partner in the Trans-Pacific collaboration and communication. It is nearly a full century since Tsinghua was founded on American Boxers' Indemnity Funds at the direction of the US Congress and under the supervision of the Chinese Ministry of Foreign Affairs. Tsinghua's founding vision was to build an outward-looking institution that pursued scientific knowledge in a liberal educational environment of humanitarian learning. Tsinghua sending this week to Berkeley such a large delegation is both a testament to our developing relationship going forward and a rejuvenation of long-standing ties that have been deeply embedded in a full century of US-China international relations. I'm hopeful, in short, that the interactions of the Tsinghua Week at Berkeley will lay the foundation for further communications and collaborative arrangements across the Pacific, resulting not only in improved intercultural competence, but also in enhanced educational infrastructure that will benefit all members of the Berkeley community. My thanks to President Gu for leading so many of Tsinghua's faculty and students to Berkeley. I'm honored by the special place that Berkeley holds in Tsinghua's strategic planning. I look forward to the exchange between members of the faculty and the student body between the two universities in the next 3 days. I'm elated both by the prospect of reaching into Berkeley's new frontier of knowledge across the Pacific and by the knowledge of bringing home to campus an old institutional partner of long-standing historical connections. My warmest welcome to our Tsinghua colleagues upon their visit to Berkeley.
Thank you, Chancellor Birgeneau. We now welcome President Gu Binglin of Tsinghua University. President Gu is an academician of the Chinese Academy of Sciences, and just like our own Chancellor, is a physicist. Please.
So thank you, Chancellor Birgeneau. Dear friends and colleagues, ladies and gentlemen, it is our great honor to have the Tsinghua Week at Berkeley in this lovely spring season. First of all, I would like to express our heartfelt thanks to the Chancellor Birgeneau and the Berkeley colleagues for your kind invitation, warm hospitality, and hard efforts to make this exciting events to be held on your beautiful campus. 2 years ago, we had the successful Berkeley Week at Tsinghua. The lectures by your faculties, the wonderful performance of your octet singers, and the men and the women's tennis players all gave us deep impressions. Now, we return to visit to your prestigious university with over 100 delegates of our faculty and students. It is interestingly coincident that by this April, we are stepping into the centennial anniversary year of Tsinghua University, which was founded in 1911. Tsinghua University has experienced and witnessed the social and economic changes of China and the world. Looking back into the past century, we, the mankind, have experienced the challenges of war and peace, poverty and development, industrialization and technological revolution, as well as the current widely discussed global climate change. All this makes us realize that only cooperation can help us to succeed in meeting various challenges. As leading universities facing challenges with global influence, we should study and address the key issues, organizing research and discussions, and deliver related education programs to our students who may be the future leaders. In this light, regular faculty and students exchange between world-famous universities can play an important role in the globalization in higher education. For this reason, we fully support such collaborations and academic exchange events between our two sides. As a leading university in China, Tsinghua has built up close academic relationships with many prestigious overseas partners. Among them, Berkeley, UC Berkeley, is definitely a very important one. Despite the different cultural and historical traditions, our two universities are both research-oriented, sharing common interests and objectives. We both pursue academic excellence and provide quality education to our students. As members of Asia and Pacific Rim universities, we share the same challenges facing the region and the world. This is why we are here in Berkeley, encouraging mutual understanding, developing friendship, and promoting academic relations. The Tsinghua Week at Berkeley will have panel discussion covering themes of energy and environmental issues, higher education, literature and history, poverty alleviation, development of new technologies, business innovation, and entrepreneurship, as well as other academic and administrative topics. I believe this week's events will build up a platform for sustainable cooperation between our two universities. One other feature of this exciting event is the 44 students members of our delegation. They will have extensive interaction with your Berkeley peers. Besides the tour on campus, they will perform Chinese folk dancing and music together with the octet singers. The interactive activities between the younger generation are important for our two countries, our two nations. As we all know that Sino-US relations is one of the most important bilateral relations in the world. The increase of mutual understanding of the young people and youth leaders of our two countries will help bridge the cultural gaps and improve the future bilateral relations of two great countries. Berkeley is the first international partner of Tsinghua after the normalization of bilateral relations of our two countries. In 1978, the two universities signed an agreement for academic cooperation. However, the historical relations just as the Chancellor Berkeley said. We can be traced back to even earlier years. In Berkeley's East Asia Library, there is a memorial room for Yuan Zhao, who taught in Tsinghua in 1920s and 1930s, and then moved to the United States and taught in Berkeley for 15 years until his retirement in 1962. In recent years, the exchanges and collaborations between our two institutes increased rapidly, such as Tsinghua Berkeley Poverty Alleviation Program, Tsinghua Berkeley Global Entrepreneurship Education Program, Partnership for Advanced Education Program, and Partnership for Advanced Transit and Highways. C-TRIS program between Tsinghua's Information Schools and Berkeley's Engineering School, and the IUP, Inter-University Program for Chinese Language Studies, and so on. It is no doubt that the Tsinghua Week at Berkeley will be a landmark event in the history of bilateral relations of Tsinghua and Berkeley, not because of the size of the delegation, but for the higher productive and stimulating panel discussions. 2 years ago, with the slogan of Berkeley Week at Tsinghua, is that partnership for excellence. Both of us did an excellent job in Beijing in that time. Today, with the same best wishes, let's work out an excellent and successful Tsinghua Week at Berkeley. Thank you very much.
Now, to take up the issue of extending the frontier of human knowledge and human understanding from a different perspective, we turn to Professor Jasper Rine to present Berkeley's keynote speech. Professor Rine is a professor, it's Howard Hughes Professor, and then also a professor of genetics, genomics, and development here at Berkeley. He has been on the faculty since 1982. He was formerly director of the Lawrence Berkeley Laboratory Center for human development, and then also the director of the Center for Computational Biology. Now, like Professor Xie, he's been awarded the Best Teaching Award on Berkeley campus. And among the many honors that he has received, he is a member of the National Academy of Science, a member of the American Academy of Arts and Letters, a fellow of the American Academy of Microbiology, and the American Association for the Advancement at Science. And I will ask him to deliver the title of his own talk so that it will not be mangled by a historian.
Let me start by thanking you very much for the opportunity to be here today. As the only biologist who will be addressing you during your visit here, I feel especially honored, but also challenged because I will be able to describe a very narrow slice of the biological research that goes on here at Berkeley. And I'd like to start by sort of identifying that analyzing variation in all of our genomes, all of our genetic differences, is really the defining problem in human biology for this century. What I'll tell you about today is our efforts to look for what I call the good news in the human genome. You'll understand what I mean by that by the next 24 minutes. But before I do that, let me take a few minutes to express our appreciation for the remarkable contributions that are being made to the biological sciences from the Beijing Genome Institute and other institutes of its type in China. I believe it is the largest genome institute in the world. It certainly produces more information on DNA from different genomes than any place else I know of. It is filled with the latest technology and is making a magnificent contribution to the world's effort to sequence the genomes from many people from around the world. In the next 2 years, we can expect to see tens of thousands of human genomes sequenced, and the challenge will be for us to understand what to do with it. China's Beijing Genome Institute was actually the place to sequence the third human genome, that of a Han Chinese. And I have to say, although it wasn't the first two sequences, it was much better compiled and presented than either of the first two sequences. And this represents the beginning of collaborations with Berkeley, as our own Rasmus Nielsen, one of our professors of population genetics, made a modest contribution to that. I'll also point out that that institute has been sequencing the most important genome on the planet, that of rice, which feeds, of course, half of the people on the planet. And the genomes of the 1,000 cultivars of rice will be the source of survival of our major food source to the impact of global climate change. So this is an incredibly important endeavor. And of course, the genome of the cutest charismatic macrofauna on the planet, the giant panda, was published from this institute just a couple years ago, though I was sad to see that the genetic explanation for cuteness was not able to be determined from that sequence. Now, Berkeley is not a leader in producing genome sequence information. That is not us. We do have the benefit of a local collaboration with the Department of Energy lab at the Joint Genome Institute in Walnut Creek, just 20 miles or so east of here, which uses, like the Beijing Genome Institute, the latest generation of so-called next-gen DNA sequencers to produce information on the original human genome sequence, but more recently on the genomes of bacteria, of fungi, and all of the creatures that fill out the evolutionary tree that we use to drive all of our scientific discoveries. Now, the first two people to have their genome sequenced was, of course, Jim Watson, there on the left, with his colleague Francis Crick, who first determined the three-dimensional structure of the DNA molecule, and also by Craig Venter, who is the director of the institute called the J. Craig Venter Institute. And of course, if you were the director of the institute, you get to have your genome sequenced. Now, what was learned by looking at these two sequences and the sequences that have come since then? Well, of course, DNA is the chemical nature of our genome. And what we learned was that our genome is made of 3 billion bases of information, the so-called A's, T's, G's, and C's. And I'm very happy to announce that the computer did not alphabetize it before we had a chance to analyze it. And interestingly, among those 3 billion bases, each of us differ in about 3 million places. So unless you're an identical twin, you are a rather unique individual, much rarer than 1 in a million. Now, of those differences, some of them are in the part of the genome that encode the instructions for making the 22,550 different proteins that drive human biology. And the differences in those proteins result in 2,000 differences that affect the structure of our proteins. 120 of those differences in any of us are expected to be deleterious or harmful to the function of that protein. Now, if you're getting nervous, you can relax a little bit because, of course, you have 2 copies of each of your genes, and most of these differences are found in one of them. But it is sobering to realize that 5 of these changes are expected to be lethal were you to have 2 copies of that different variant. So we have genomes which are made of many problematic genes, and our goal is to really try to understand what the impact of all of that genetic variation is and what we can do about it. Now, I'll focus today on the portions of our genomes that encode proteins, and half of the proteins are required to drive the chemistry of life, and the wiring diagram of human metabolism, a simplified view of that, is shown on the left. Each of the dots represents a chemical that can be found in our cells, and each of the line represents a chemical transformation from one to another that is catalyzed by one of our protein enzymes. Now, to experience the nightmare that all of our biochemistry students experience, let me fill in that slide with more detail. And the important point here is not the detail, but the recognition that we know an enormous amount about the enzymes and proteins that drive our biochemistry, but very little really about the impact of genetic variation on that biochemistry. So how do we analyze this? Well, it's really pretty simple. We start with the sequence of genes, and then there is a table called the genetic code, and if we simply look at all of the letters in our genome and translate them with that code, we can figure out what the sequence is of individual proteins. So there's the sequence of a protein written out in the code in which each letter represents a different amino acid. Now, although no individual or no computer on the planet can figure out what the structure of that protein will be, the protein on its own, because of the chemistry of the amino acids, can fold itself into its three-dimensional structure. And this three-dimensional structure is what gives each of our 22,550 proteins its unique properties, its unique roles. And because the structure comes from the sequence, and the sequence is determined by the gene, then changes in our genes can change the structure of those proteins in ways that we want to learn more about. Now, one of the challenges has been brought clear in just the last couple weeks with the publication of a paper by two groups in Seattle which described the sequencing of the genes in a father and a mother and both children to try to track down the gene responsible for an inherited disease. Without going into the details of the disease, the thing I'd like to point out is that that simple experiment with 4 people produced 20 terabytes of data that needed to be analyzed. Now, we biologists have long felt inferior to our physics colleagues like George Smoot and to the economists of the world that deal with mountains of data, but no longer. We have more data than we can handle. So what is Berkeley's position in all of this? Berkeley is a leader in what I like to call next-generation biology, the biology that is driven by the discoveries in our genomes from next-generation DNA sequencers. I've told you about the torrent of data from the next-generation sequencers that we suffer with, and our first line of defense is computation. I'll tell you about our approach to that, and it is the computation which then provides the information that drives our discoveries in genetics, in biochemistry, structure determination, and model organism research, which then allows us to interpret the function of human genetic variation. I'll give you just a Cook's tour of some of the efforts at Berkeley before I focus on one project aimed at finding the good news in the genome. We have a Center for Computational Biology made up of 33 faculty from 9 different departments, which are remarkably talented. The center is presently led by Lior Pachter, a professor of mathematics and molecular and cellular biology. And rather than telling you what each of these 33 faculty do, I'll just list a subset of the recognitions that they've received. You can see it's a pretty illustrious group. We also have at Berkeley what I believe is unique in the world, a place that we call MacroLab. It is a facility that creates the capacity for high-throughput protein biochemistry. This was driven by the needs of our crystallographers to produce lots of different proteins, and what I'll show you is just a subset of the equipment that make up MacroLab. The equipment is utterly unimportant. You can buy that any place, but what is really important is the integration of all of this equipment to create a unique opportunity for the analysis of hundreds of different proteins a week, thousands of different proteins a year, in an organized research structure that could be easily cloned and exported to other venues. What do we do with all of these proteins? Well, among the things that we do is to use a world-unique facility at the Lawrence Berkeley Lab called the Advanced Light Source, in which different beams of synchrotron radiation have been allocated for determining the structures of different proteins. We first crystallize proteins, then we mount those crystals in the middle of an X-ray beam shine high-intensity, short wavelength light through them, and the diffraction pattern lets scientists like James Berger and his student determine the structures of different human proteins. We have 12 world-famous protein crystallographers on campus, and people from around the world come to Berkeley to use the advanced light source on their samples. So that's a quick tour of the kinds of things that we do for next generation biology. And you have probably heard of the central dogma of molecular biology, but alas, genome analysis has not a central dogma, but a central dilemma. And the central dilemma is paraphrased by Mark Twain, one of the most famous American writers, when he wrote that everyone complains about the weather, but nobody does anything about it. Well, with respect to the genome, we all have problems in our genome, as I've shown you, but is there anything that we can do about it? So the nature of the challenge is that among the 3 million differences, we use a word called polymorphisms to represent those differences, we can presently interpret the significance of less than 1% of those. The $1,000 human genome sequence is virtually here with us, so many of you will be able to purchase the sequencing of your own genome, but what will we do with it? And the problem with genome information is that most personal genetic information is perceived as bad news. You could have a test today that would give you 50% certainty that you will have Alzheimer's before you're 50. For some reason, people do not embrace the opportunity to have that test. You could have other tests that would tell you about your predisposition to other terrible things. People tend not to want to know that. So the question for today is whether we can create a market for personal genetic information that is good news. So what would this good news be? Well, it should be non-frightening, actionable, safe, inexpensive, and effective for improving the quality of your life and health. So that's what I'll tell you about today, which is our efforts to find that good news. And the place that we've looked, and I think the best place to look initially, is in the area of the interface between nutrition and human genetic variation, because that's the part of our environment which we can most easily alter as need be as we learn from our genes. The project that I'll tell you, as so often happens at Berkeley, is inspired by another of my colleagues, in this case, my good friend and colleague, Bruce Ames. 40 years ago, Bruce made a remarkably obscure discovery. He discovered that mutations in bacteria that you can think of as genetically inherited diseases of bacteria could be treated by giving bacteria larger doses of vitamins. So, avoiding even a shred of creativity, I asked, do such mutations exist in humans? Now the question really is, how much vitamins do we need, and does genetic variation impact that? I was surprised to discover that frankly, we don't have a very good idea about how much we need in terms of vitamins and minerals. As you look at this table, you'll discover there are big discrepancies, and you'll find that if you go from France across the channel to England, you no longer need riboflavin or niacin based upon these numbers. So why is our science so unclear on things that are so important? It's largely because these values were determined during World War II based upon the governments around the world concerned about starvation and malnutrition and famine, which often happens in wars. And experiments were done on conscientious objectors to the draft to figure out what you could starve them for before you got pathology. So we know a great deal about how to keep 18 to 35-year-old Quaker males healthy with respect to nutrition, but for old guys like me, for women, and for others, we don't have a very good idea. Furthermore, all of these values are based upon the assumption that we are all genetically the same, and we really are not. So I'll begin our discussion here with folic acid, a very important vitamin, which you get from leafy greens, and in times of famine and food shortages, you don't get enough of that. Folic acid is really important for all kinds of reasons that I'll show you, and it's particularly important if you're a woman. So here's the structure of folic acid in its chemical language, And if you're a woman with 2 X chromosomes, you need twice as much folic acid for proper health as for men. And I'll tell you why in just a few slides. So our question is there genetic variation among us which influences how much vitamin we need for human health? I'll show you one result having to do with a human enzyme called methylene tetrahydrofolate reductase, which you now understand why I'll call MTHFR. There is a very common mutation in the gene for that enzyme that changes a C at position 677 to a T that results in a substitution of alanine A for valine at position 222 in the protein. Now we know that this mutation has a frequency in humanity of 0.3. So what that means is that 42% of the people on the planet are carriers for this mutation, and 9% of the people on the planet have 2 defective genes with this mutation. So it's a sizable chunk of the world. We know that this mutation results in a decrease in the activity of that enzyme. The enzyme is unstable because of the mutation, but you don't have to worry if you eat enough folic acid. If you don't, you end up with a condition called homocystinemia, and homocystinemia is a risk factor for strokes, thromboses, and various cardiac events that you don't want to have any personal familiarity with. So 9% of the population needs extra folic acid because of this one mutation. So the question is, how many more mutations like this are among us? There are 643 vitamin and mineral-dependent enzymes that drive human biochemistry, and if we learn enough about them, can we actually individualize metabolic tune-ups that will help all of us? So the question is, how can we set about discovering these? The discovery of the A222V mutation in humans took about 20 years of research and almost 500 research papers to sort of nail that down. That's pretty slow and inefficient. So we've asked whether or not we can do this in a more efficient way. And the way that we do this is to use yeast, the yeast that makes bread, beer, and wine, as a little experimental platform to test the function of human genes. That probably sounds crazy, but let me explain it to you. First of all, we have to consider that with 7 billion people on the planet and a mutation rate of 10 to the minus 8th per base per generation, that every generation, every position in our genome is mutated de novo in at least 30 different people, okay? But each person has 3 million genetic differences, and so if we see a difference between you and me, the question is which of these 3 million differences is responsible for the difference that we notice. So what we do is to take individual human genes and all of the variants out of that variable genome of ours and plant it into the same organism so that we can compare directly one change versus another. It involves very rudimentary and easy genetic engineering. My colleague and I, this is Nick Marini, my colleague, produce specialized strains of yeast in which we turn on a human gene for a human enzyme in a cell in which we have removed the yeast's own version of that gene. We've also removed the yeast's ability to make its own vitamins, and hence it has only the amount of vitamin that we give it. We then test the function of human genes and the variants in the world's simplest, cheapest, and easy experiment. It's called a growth curve. We simply inoculate these strains of yeast into medium and measure the change in cell number as a function of time, depending upon how much vitamin or cofactor we put in the media. So this is normal growth rate, slow growth rate, and low growth rate. Now I'm going to show you next 2 slides of data. If your eyes kind of glaze over and you get sleepy, you are normal. But if your eyes are still awake at the end of that, you have a good career option in biology, so bear with me. Our first experiment is to ask, can we recapitulate in yeast what is known of that very common human mutation? So if I could direct your attention to the upper graph, I will show you the growth curve of yeast in brown, powered by its own version of the MTHFR gene. We express in blue here the normal human enzyme at a level that doesn't let it grow at the fully high rate to give us the capacity to see increases or decreases. And now we express that common variant of the enzyme, and you can see that it's growing at about half the rate, just like half of the activity that this enzyme has in humans. If we decrease the concentration of the cofactor from 50 units to 25 units, just twofold, suddenly that variant is no longer functional. So this calibrates yeast to teach us about human genetic variation. We then use that assay, sequence thousands of genes in lots of people, and what we find is remarkable. In healthy people, we find many variants that affect the function of the enzyme. Here's one that doesn't work very good even under favorable conditions. Reduce the cofactor, it's essentially dead. Here's some that look good until the cofactor concentration goes down, and we find some neutral ones. We can ask where those changes are on the three-dimensional structure of the protein, and the answer is frustrating. They're scattered all over. So it doesn't look like we've got a very good chance to solve computationally what it takes to make a cofactor or vitamin remedial mutation. We may have to do this by experimentation. Folic acid is also incredibly important for avoiding birth defects. Neural tube defects, one of the three most common birth defects, the type spina bifida and anencephaly, have a frequency in this country of about 1 in 1,000 live births, higher in some other countries, and they are folate preventable. This is why women are advised by their OB/GYN doctors to take twice as much folic acid, because if you have elevated levels of folic acid before becoming pregnant, your risk of a neural tube defective pregnancy goes down. And in 1998, the US government demanded the fortification of grains and cereals with folic acid for that reason. The two other most common human birth defects, cleft palate and congenital heart defects, are also folic acid sensitive. So we're dealing deeply now with folic acid variation in samples provided to us by children born with different birth defects to try to understand the genetics of that. We're collaborating with the California Birth Defects Monitoring Program and the Children's Hospital of Oakland Research Institute to try to figure out the genetics of neural tube defects. It's presently unknown, but the leading hypothesis is that there are genetic defects in folate utilization that underlie this. So we've sequenced and we're measuring the function of these genes from, the DNA is from blood spots that are taken on all children born in this country. We've sequenced 18 genes in 250 kids born with birth defects and 250 matched controls. We have found 774 mutations, and 126 of those affect the structure of the proteins in which the mutations reside. And we're presently trying to combine our knowledge of the biochemistry and the metabolism and which mutations affect which enzymes to try to see if we can determine the cause of that. So I'll close with the lessons from next generation biology. The first lesson is that it is literally like drinking from a fire hose. The data comes at us in enormous paces. I hope I've shown you that personal genome information can be useful and not frightening. You shouldn't worry if you find out that you need a little more vitamin B6 than your neighbor. I've shown you that nutritional requirements vary and that vitamins can fix some mutant proteins. And that raises the interesting question about whether we can design drugs that fix other broken proteins. I think we're just seeing the tip of the iceberg here with nutrition. We can also ask whether genetic variation impacts other things like perception and cognitive functions. And of course we know that it does because 7% of males are colorblind due to genetic variation. But there's new data all the time that take us deeper and deeper into the impact of genetics on our own mental functions. Just 2 weeks ago, this very interesting paper was published that teaches us that your sensitivity to pain can be explained by variation in a single locus. So I think as we'll see in the coming years, there's virtually all aspects of human biology that will be affected by variation, and that as we understand our own variation, we will understand our own biology more deeply. We've had help from a number of people on various aspects of this project, almost all of whom are local, and thank you for your interest and attention.